This is the sixth video of the fundamentals of biology, and today we're going to be going over photosynthesis. So where does photosynthesis occur? Photosynthesis occurs in autotrophs, or organisms that are able to sustain themselves without other organisms. So this includes things like plants and also algae. And within autotrophs, a photosynthesis will occur within the mesophyll, which is a part of the leaf, and is the interior tissue. And inside the mesophyll are chloroplasts. About 30 to 40 of them are in each mesophyll cell, and it contains chlorophyll, which is a pigment that makes the leaves green. And photosynthesis is divided into two steps, the light-dependent and the light-independent reactions. And the light-dependent will occur in the thylakoid membranes, whereas the light-independent will occur in the stroma, or the outside section of the chloroplast. So just like cellular respiration, photosynthesis is also a redox reaction, or a reduction/oxidation reaction. So just to review, reduction is the gaining of electrons, which means you have a more negative charge, whereas oxidation is a losing of electrons. And you can remember this through oil rig. So if you look at the equation for photosynthesis, it is basically the opposite of cellular respiration. So the carbon dioxide will be reduced into glucose, whereas water will be oxidized into oxygen. And it will also require an input of energy, which in this case is light energy. And because of this, respiration is endergonic and uses energy. So now let's talk about light absorption and how plants are actually able to use light energy. So pigments are things that are able to absorb light photons, and they best absorb different wavelengths of light, usually shown through nanometers. So light is both a wave and a particle, and these particles come in the form of photons, and the waves come in the form of wavelengths. And the absorption spectrum will show how different wavelengths affect the absorption of light. So you can see in this absorption spectrum on the top for different pigments, you can see that a lot of it will be concentrated around here and here in terms of what will be best absorbed. On the other hand, action spectrum will be the wavelength versus the rate or effectiveness of that light. So it will also look similar to the absorption spectrum because it is the most effective when it can absorb the most light. And some pigments to remember for photosynthesis is chlorophyll A, which is the primary pigment used for plants in capturing light, and chlorophyll B in carotenoids will broaden the spectrum for photosynthesis. So they expand this light spectrum in order to make it easier for the plants to absorb light. And you can see that violet blue light as well as red light will be the most effective usually. So the first step of photosynthesis is the light reactions, and they are called light reactions because they depend on the presence of light in order to work. And first we have to talk about what photosystems are. Photosystems are found on the thylakoid membrane and they are embedded within it. And they consist of a reaction center complex, which is surrounded by light harvesting complexes. So the reaction center is this thing in the middle, whereas the light harvesting complexes surround it. And as a light photon hits a pigment molecule, which in this diagram is shown with the green circle, the energy is transformed and it bounces around these pigment molecules until it attaches to a pair of chlorophyll A molecules within the reaction center complex. And as a result of this, the electron will be excited and it will be accepted by a primary electron acceptor. And there are two photosystems used in the light reactions, known as photosystem 2 and photosystem 1. And photosystem 2 will actually occur first, whereas photosystem 1 will come later. The 1 and 2 simply just tell you the order in which they were discovered. And photosystem 1 is also called P680, which is the light that it can best absorb, 680 being the wavelength in nanometers. And photosystem 1 will be P700 based on the best absorption wavelength. In the light reactions, there are there will be a linear flow of electrons. So when a photon first hits the photosystem 2, the electrons are caught by the primary acceptor, which we talked about earlier. And after that, water over here will be split in order to replace that electron that we just used. And H plus ions will be released into the thylakoid space, and the O2 will be a byproduct of that water. And the electron that we just talked about, they will travel down something called the electron transport chain, or the ETC. And they will pump protons into the thylakoid space via something called the cytochrome complex. So this will be the cytochrome complex. And the buildup of H plus ions inside the thylakoid and the proton gradient will drive something called chemiosmosis, as protons will move into the stroma via ATP synthase, which will create ATP for the Calvin cycle. So if you want to learn more about chemiosmosis, you should check out the video about cellular respiration. The next step is for the electrons to pass through photosystem 1, but instead of the electron traveling down the ETC, it will be used to reduce NADP plus to NADPH. And NADP plus is going to become an electron carrier, and that basically just takes the electrons and carries it to the next step of the photosynthesis, and will be used in the Calvin cycle. So usually electrons will travel through one way, but sometimes it will go through a cyclic flow. And that basically means that after the electrons in the photosystem 1 are caught by the primary acceptor, they will travel back to the ETC in order to produce ATP. And this will happen by pumping more protons. And in this, NADPH will not be produced because it will not be linear flow into the NADP+. And the purpose of this is to protect cells from light-induced damage. The next step is called the Calvin cycle. It is light independent, which means it does not rely on the presence of light. So the first step is called carbon fixation. What happens in carbon fixation is an enzyme called rubisco will attach 3 carbon dioxide molecules to 3 RUBP molecules, and ATP will make 6 3-carbon molecules that are excited. The next step is called reduction, and using 6 NADPH molecules, 6 G3P molecules are created. And after that, 1 will be released as half of the glucose molecules, and 5 will be kept in the cycle. And finally, we have to regenerate the RUBP molecule, and using ATP, the carbon dioxide acceptor is regenerated. So just as a note, 2 G3P molecules will be required to make 1 glucose since there are only 3 carbons and 1 G3P. So therefore the cycle needs to run twice. So if we want to just follow the number of carbons in the cycle, we can see that we will initially start with 3 carbon dioxide, which means there are 6. And if you add that to the 3 RUBP molecules, which have 5 each, you will have a total of 15 plus 3, and that will be 18 carbons. So that makes sense because this is a 6 carbon molecule, and there are 3 of them, so that is 18. And once they split apart, it will become the same, basically, because you'll just have 2 of the same things. And after, you're not taking any carbons out, but then you will take 1 G3P out, which will remove 3 carbons, so that becomes 15. And that's how you have 15 over here. So as a general overview of photosynthesis, the light reactions will only occur in the presence of light and water, and it will create O2 as a byproduct, and ATP and NADPH will be used in the Calvin cycle. And the Calvin cycle will use carbon dioxide to produce sugar, and it relies on ATP and NADPH from the light reactions, but it will regenerate ADP and NADP+ for light reactions. So thanks for watching this video. This lesson covered absorption of light, the light-dependent reaction, and the Calvin cycle.